A very warm welcome to all of you guys. Welcome back. Welcome back in another video tutorial for microbiology with Chagni. So today we will going to talk about another important thing on our carbohydrate metabolism, that is the Bifid Shunt. So it was discovered by scientist Suzuki et al, and he used this concept of Bifid Shunt to explain how organisms like Bifidobacterium very cleverly combines phosphoketolase pathway that we have already discussed and pentose phosphate pathway to yield one extra ATP from 2-glucose. Okay, if that bifidobacterium organism will going to use bifid shunt, remember it will got the opportunity to yield 2.5 ATP per glucose and hence 5 ATP from 2 glucose, but usually it only got the opportunity to yield 4 ATP. Therefore, one extra ATP if these organisms like bifidobacteria will going to follow this bifid shunt. So let's get started our discussion on what is this bifid shunt is. So here I have already explained you what I have written here. So the main funda of this Bifid shunt is an enzyme which is actually thiamine diphosphate dependent enzyme. And this type of enzyme usually present in all organisms and they involved in various type of reactions in various important metabolic pathways. So thiamine diphosphate dependent phosphoketolase is the main enzyme here which will going to play a key role in this Bifid shunt. They, although phosphoketolase play a important role in central metabolism of heterofermentative bacteria, that is our phosphoketolase pathway we have already discussed, but in that time remember it was thiamine pyrophosphate dependent phosphoketolase, right? And here it is thiamine diphosphate dependent phosphoketolase. That is why they are quite different. Remember, due to the thiamine diphosphate dependent phosphoketolase have the extra ability to react on two substrate. Okay. On the other hand, thiamine pyrophosphate only have the opportunity to react with only one substrate. That was the xylulose 5-phosphate, right? So that is the thing. So here we will going to discuss the bifid shunt that is occurring in bifidobacteria. Okay. So without any further delay, we will going to use the diagram to understand the Bifid shunt. So let's move to the diagram. So this is the diagram we will going to use. So here it all starts with glucose, two glucose molecules. Okay, so two glucose molecules will be converted into 2 fructose 6 phosphate, and we all know what are the two enzymes that converts glucose to fructose 6 phosphate. First hexokinase converts glucose to glucose 6 phosphate, and then from the help of isomerase we will get fructose 6 phosphate, right? Now on one fructose, one xylulose fructose 6 phosphate phosphoketolase, the full form of this XFPK is the xylulose 6 phosphate xylulose 5 phosphate slash fructose 6 phosphate phosphoketolase will going to act. Okay, so what I have told you, first 2 glucose will be converted into 2 fructose 6 phosphate, and on 1 fructose 6 phosphate xylulose 5 phosphate slash fructose 6 phosphate phosphoketolase will going to work. Although here this enzyme will also get the help of PI that facilitates phosphorylated cleavage, yielding erythrose 4 phosphate which is a aldose and acetyl phosphate. And the most interesting thing here is, here in this phosphoketolase mediated cleavage there will be removal of water. That is the difference between thiamine pyrophosphate dependent enzymes phosphoketolase which participates in the our only phosphoketolase pathway, and this thiamine diphosphate dependent phosphoketolase which are participating here. Okay, this second type of phosphoketolase have the ability to yield water during the phosphorylated cleavage. Understood? So now this erythrose 4 phosphate and fructose 6 phosphate in presence of transaldolate will be converted into glyceraldehyde 3 phosphate and pseudoheptulose 7 phosphate. I have already explained you how you can remember that how transaldolase and transketolase work. If you don't know, I would like to recommend you to go and watch the pentose phosphate pathway video. Then it will be easier to you for understanding that case. Okay, so still I will going to explain you here. That is, remember in transaldolase, when transaldolase works, it will going to snatch 3 carbon containing molecule from fructose 6 phosphate and making it a aldose. Okay, again I am repeating, when transaldolase works, it will going to snatch 3 carbon from fructose 6 phosphate and making this fructose 6 phosphate into a aldose of 3 carbon containing molecule. So 3 carbon containing aldose is glyceraldehyde 3 phosphate, we all want or we all know, and the 3 carbon that it have snatched from fructose 6 phosphate will be transferred to the erythrose 4 phosphate and making it a ketose. So 7 carbon containing ketose is very important MCQ question from the biochemistry, that is pseudoheptulose 7 phosphate. Okay, now they will again going to react in presence of transketolase. So here the idea is transketolase will going to snatch 2 carbon from the higher carbon containing ketose. Okay, so if it snatch 2 carbon containing molecule from pseudoheptulose 7 phosphate, it will become a aldose of 5 carbon containing. So aldose of 5 carbon containing is ribose 5 phosphate, and now the 2 carbon that it have snatched from pseudoheptulose 7 phosphate will be transferred to glyceraldehyde 3 phosphate, making it a ketose of 5 carbon, right? That is xylulose 5 phosphate. Now what will happen? This ribose 5 phosphate in presence of ribose 5 phosphate isomerase converted into its ketose form, that is ribulose 5 phosphate. Then this ribulose 5 phosphate will going to converted into xylulose 5 phosphate in presence of epimerase. Hence we get 2 xylulose 5 phosphate, right? Now again xylulose 5 phosphate fructose 6 phosphate phosphoketolase will come, and here 2 pi will go into help it and yielding to glyceraldehyde 3 phosphate, to acetyl phosphate and 2 H2O. Again you can see here there is another dehydration, right? Now one thing I hope you have noticed, that in both the cases we are getting aldose, right? One erythrose 4 phosphate, another one is 2 glyceraldehyde 3 phosphate. Now this 2 glyceraldehyde 3 phosphate will going to enter the EMP pathway, giving us lactate, and this 2 acetyl phosphate will going to combine with another acetyl phosphate that we have get during the first action of xylulose 5 phosphate fructose 6 phosphate fructose 6 phosphate phosphoketolase and converted into 3 acetyl phosphate. Now this 3 acetyl phosphate in presence of acetate kinase will going to convert it into acetate. That means the phosphate group is removed, right? So that is the idea. That is the idea of our Bifid shunt. Okay, so you can see glucose will be converted into 2 fructose. 1 fructose will going to cleaved with the help of this phosphoketolase, that is thiamine diphosphate dependent phosphoketolase, giving us erythrose 4 phosphate, acetyl phosphate, and importantly water. And this erythrose 4 phosphate and fructose 6 phosphate will going to enter in the pentose phosphate pathway. I hope you can remember these reactions are belongs to the pentose phosphate pathway. Then we will get 2 xylulose 5 phosphate that again going to cleaved by phosphoketolase, giving us glyceraldehyde 3 phosphate acetyl phosphate, right? And now these 2 acetyl phosphate and 1 acetyl phosphate together becomes 3 acetyl phosphate, and this 3 acetyl phosphate will be converted into 3 acetate by acetate kinase. Okay, so that is the idea. So that is the idea how bifidobacterium yields 1 ATP extra from 2 glucose molecule. Okay, so that is the idea of our Bifid shunt. So I hope this video will be helpful for you, and I hope you have understood the concept very well. Another important thing that I have forgotten to mention, that is this xylulose 5 phosphate fructose 6 phosphate phosphoketolase have the ability to react with this fructose 6 phosphate as well as xylulose 5 phosphate. This specific tendency of this phosphoketolase, which is thiamine diphosphate dependent phosphoketolase, is known as dual substrate specificity. Okay, it is very important. You have to remember that is dual substrate specificity. So that's the idea. So I hope this video will be helpful for you. Thank you.